In this lesson, we look at DNA as an individual identifier. We'll also look at the uses and abuses of DNA fingerprinting. So in another time, Colin Pitchfork, a murderer and a rapist, would have walked free. But in 1987, he was captured and convicted, betrayed by his own DNA. And he's now serving 2 life sentences in prison. Pitchfork's downfall began when he raped and murdered 2 15-year-old high school girls in a small village in England in the 1980s. The police thought they had their perpetrator when a man confessed, but only to the second murder. He denied any involvement with the first murder, which perplexed the police because the details of the 2 crimes strongly suggested that the same person committed both. At the time, British biologist Alec Jeffreys made the important discovery that there are small pieces of DNA in human chromosomes that are tremendously variable in their base sequences. In much the same way that each person has a driver's license or a social security number that differs from everyone else's, these DNA fragments are so variable that it is extremely unlikely that 2 people would have identical sequences at these locations. Thus, a comparison between these regions in a person's DNA sample and in DNA-containing evidence left at a crime scene would enable police to determine whether the evidence sample came from that person. Jeffreys analyzed DNA left by the murder rapist on the 2 victims and found that it did indeed come from a single person, and that person was not the man who had confessed to one of the crimes. The original suspect was released and has the distinction of being the first person cleared of a crime through DNA fingerprinting. To track down the criminal, police then requested blood samples from all men in the area of the crimes who were between 18 and 35 years old, a practice that many viewed as an invasion of privacy. The police collected and analyzed more than 5,000 blood samples. This procedure led them to Colin Pitchfork, whose DNA matched perfectly the DNA left on both victims, and ultimately was the evidence responsible for his conviction. You see him here in the image. He almost slipped through, having persuaded a friend to give a blood sample in his name. But when the friend was overheard telling the story in a pub, police tracked down Pitchfork to get a blood sample. DNA fingerprinting is now used extensively in forensic investigations in much the same way that regular fingerprints have been used for the past 100 years. But regular fingerprinting is limited in its usefulness for many crimes because often no fingerprints are left behind. DNA samples are frequently left behind, though, usually in the form of semen, blood, hair, skin, or other tissue. As a consequence, this technology has been directly responsible for bringing thousands of criminals to justice and, as we saw at the beginning of this unit, for establishing the innocence of more than 300 people wrongly convicted of murder and other capital crimes. Let's next examine how DNA fingerprinting is done, why it is such a powerful forensic tool, and why it's not foolproof. The DNA from different humans is almost completely identical. More than 99.9% of the DNA sequences of 2 individuals are the same because we're all of the same species and thus share these traits. Even so, in comparing 2 individuals' genomes of 3 billion base pairs each, 0.1% difference still translates to about 3 million base pair differences. These differences are responsible for the fact that all individuals have their own unique genome. The lone exception? Identical twins, whose DNA is exactly the same. Thus, when we are trying to evaluate whether the DNA from a crime scene matches that from a suspect, the analysis focuses on the parts of our DNA that differ. There are thousands of these highly variable regions in the human genome. Among these thousands of variable regions, one particular type is used for the determination of a person's genetic fingerprint. These regions are called STRs. This stands for short tandem repeats, and they're characterized by a short sequence, which is commonly 4 or 5 nucleotides that repeats over and over within a region. The number of repeats is what varies among individuals. An individual, and we'll call her in this case individual A. Individual A has 2 copies of each chromosome, one from her mom and one from her dad. At one STR region on chromosome 2, for example, the number of times the sequence repeats is likely to differ on the maternal and on the paternal copies of that chromosome in individual A. The sequence may repeat 3 times on the maternal copy of a chromosome 2, and it may repeat 14 times on the paternal copy. In such a case, individual A is said to have 2 different alleles for this STR region, 3 and 14. In contrast, in individual B, for the same STR region on chromosome 2, the sequence may repeat 5 and 11 times. You can see that depicted here in the illustration. For an STR region within the human genome, there are typically about 10 different alleles that occur within a population, and each is separated by about 10% of all individuals in a population. So the likelihood that 2 individuals carry the same 2 alleles is about 1 in 100. This is unlikely, but given enough people, many are likely to carry the same alleles. If 2 different STR regions are analyzed, the likelihood that 2 individuals have the same 4 alleles is 1 in 10 by 1 in 10 by 1 in 10 by 1 in 10, or 1 in 10,000. This is much rarer, but still would lead to multiple individuals within the same large city carrying the same 4 alleles. The real power of DNA fingerprinting comes from simultaneously determining the alleles an individual carries, that is, their genotype, not simply at 1 or 2 STR locations, but at 13 different STR locations. This is the number used by the FBI in constructing DNA fingerprints in the United States, and it makes the probability that 2 individuals would have exactly the same genotype extremely low, as you can see in the image here. For each STR locus analyzed, an individual's genotype is determined by using PCR to amplify that region, then measuring the length of the STR region using a technique called electrophoresis, which allows researchers to see how long a stretch of DNA is. The length of the region can then be used to determine the number of times the STR is repeated. For a single STR region, an individual's genotype is expressed by 2 numbers, reflecting the number of STR repeats in the copies inherited from the mother and from the father. And a person's full DNA fingerprint is a string of 26 numbers that includes the 2 numbers for each of 13 STRs. In court, a suspect's genotype might be compared with a DNA fingerprint obtained from evidence found at the crime scene. DNA samples from different people will produce different 26 number fingerprints, whereas different samples of DNA from one person will have exactly the same genotype. In the end, despite universally accepted methods, DNA fingerprinting still is not foolproof. Numerous incidences of human error, accidental as well as intentional, have been documented, ranging from mislabeled test tubes to tissue from a suspect being added to evidence from a crime scene. So we should not blindly draw conclusions solely from this one type of evidence. Nonetheless, DNA fingerprinting is an increasingly valuable tool for law enforcement. The FBI, for example, has reported that nearly 1/3 of their suspects are cleared immediately by DNA testing, and that many more criminals, because of DNA fingerprinting, now plead guilty to the crimes that they have committed.